Good day, everyone. Welcome. I'm Jack Van Horn from the University of Virginia. Please allow me to welcome you to this edition of our 2020-2021 Foundations of Biomedical Data Science Seminar Series. This lecture is supported by the UVA Integrated Translational Health Institute of Virginia, the UVA Brain Institute, the College of Arts and Sciences, the School of Data Science, and by a grant from the National Institutes of General Medical Sciences. It is my honor and privilege today to welcome our speaker, Dr. James Morris from the University of Virginia. Dr. Morris serves as an associate professor of psychology and is director of the graduate admissions program here at the UVA Psychology Department. Work in his laboratory is focused on how the interactions, thoughts, and beliefs and feelings of others are represented in the brain, processes collectively referred to as social perception. Techniques employed in the laboratory include functional magnetic resonance imaging, event-related potential recordings, and molecular genetics techniques. Most of the work is concerned with building basic neurobiological models of social function, but also explore the neural basis of social deficits associated with autism spectrum disorder. Additional work seeks to examine how individual difference in social perception may be related to epigenetic mechanisms. Two broad projects currently going on in Dr. Morris's Social Neuroscience Lab involve a longitudinal investigation of the neural development in preschool children, as well as a project in healthy adults aimed at identifying variability in neural systems that is predictive of differences in social capacity. Finally, in order to appropriately consider the promise of brain mapping techniques in drawing conclusions about individual differences, Dr. Morris and his laboratory seek to identify new approaches which move from simple associations to predictive frameworks, allowing for better causal inferences and less false positive findings. Dr. Morris's lecture today is entitled Functional Imaging of Individual Differences in Social Cognition. As always, we are streaming this lecture live and for recording via YouTube. If you're watching via YouTube, thank you so much for joining us. And also our specially selected 2020-2021 Biomedical Data Science Innovation Lab participants are encouraged to submit questions for Dr. Morris via the chat feature in their Zoom sessions. I will synthesize these questions and ask them on your behalf during the last 10 minutes or so of the lecture. And with that, welcome, Jamie. We are so excited to hear from you today.
Thank you. I appreciate the introduction, and I'm happy to tell you a little bit about the work in my lab. I've kind of made this somewhat general in terms of some of the bigger themes that have gone on and some of the directions we're moving in that I think address some of the more important topics going forward when considering individual differences in social cognition, social perception, and particularly with the use of neuroscience methods to look at these factors. I've been using this slide for years. It's still relevant. It gives a little bit of like a little postcard of psychologically what I'm interested in. I am in a department of psychology. I am a psychologist by training. So I do have an interest in one sort of domain area, although the work becomes pretty broad fairly quickly. When I talk about what I'm interested in terms of my domain of research, I talk about this idea of social perception, which isn't sort of a concrete definition. It's really sort of a catch-all for a number of skills that fall under an umbrella. And you can think of it as a set of skills centered around our ability to identify other social entities and to make inferences about the dispositions, actions, goals, and intentions of others. These are things that humans reflexively do. They follow a pretty strict developmental course as children are growing up. And they're important for the way that we build social relationships, for the way that we build teams, or the way that we build close relationships, romantic relationships. They help us in terms of promoting social proximity. It allows us to essentially get to know one another and to get to understand one another, which allows for deeper sort of connection. And this connection obviously is important from a nourishment and health standpoint. And it really sort of colors what it means to be human in a number of ways. But I look at the very basic portions of this, again, going back to things such as identity. So how do we understand who people are based off of facial characteristics, people we've met versus people we haven't. And then how do we take simple cues, such as gaze cues or simple biological motion cues to understand what other people are doing and what they're thinking. And these are things that we're really good at, but they are also things that deficits exist in our population, and deficits are often associated with social and emotional sort of disorders, so developmental disorders and psychiatric disorders. So there's a lot of importance that gets placed through them. Now, in order to sort of take you through the way that I've come to understand this and the way that I've come to understand the neuroscience of this, I'll take you through the way that the field has come to understand social perception and the way that we have relied on the technologies of the time and the way that we have then used these technologies to make our inferences and the goals of our particular studies. Now, what you have with fMRI, so fMRI obviously is a technique that has been dominant in cognitive neuroscience research since about the late 90s. The earliest studies were done in the early 90s, but in terms of actually getting a critical mass of literature, you started to see a big uptick in the late 90s into the early 2000s, into now where it's it's ubiquitous. It's all around, new publications every day. Obviously some real sort of core areas, et cetera. One of the things that was a technique that you would see very early in fMRI research is what I would call studies of category specificity. And this is really specificity with regards to the visual system. So you can think of the visual system, you can think of really basic things like gradients and checkerboards, and how primary visual cortex is very dedicated to certain visual stimuli, which is very true when you get into primary visual cortex. When you get into the secondary visual cortices, you start to see specialization for more complex visual items. And you can see some level of patterning or category specificity. And fMRI studies have established that there are certain patterns that you can associate with certain stimulus classes. For instance, faces. So in 1997, Nancy Kamlisher used this term fusiform face area to describe some of her work and some of the other work, most notably coming from places like Greg McCarthy's lab, looking at the pattern of response in this fusiform gyrus area when looking at faces compared to other stimulus classes. For instance, things like letter strings, which also show a clustering of activity in most people's brains. Also during this time later, a couple years later, you would see reports of things like places, which again, work out of Kamlisher's lab. What they would do is they'd functionally characterize the response and then use a name so that they can go back and test these areas later. In this case, it's called the parahippocampal place area. Even to the lateral surface here, where people had described activity towards these headless bodies, which I could give a different talk about what we think is going on here. But essentially just that there are some areas too that seem to have odd sort of characteristics in terms of the preference for response, in terms of how strong they respond to different visual classes of information. And then finally, one that I've worked on a lot, biological motion perception. Kevin Pelfrey, who's a professor here at UVA now, an expert in neurobiology of autism spectrum disorders, also did a lot of the seminal work in fMRI with biological motion perception. And he did a lot of work here, and I did some work with him as well in my training on biological motion characteristics in the superior temporal sulcus area. And essentially what we mean by this is an area that, unlike general coordinated motion, which tends to be carried out and processed back in more primary visual cortex, this is a secondary, more of association type of cortex that seems to be activated more by biological motion. So motion made by humans in terms of point light walkers or other sorts of animations that we can see. Okay, now I want to actually go backwards in time. A lot of the work that we had noticed from early fMRI work really came out of other domains. One domain would be EEG, which is electroencephalography, or ERP type of work, event-related potential work, and that's often from the scalp. But we also, under rare conditions, have chances to record directly from the human brain. And work by my former mentor, Greg McCarthy, Truett Allison, the Spencers at Yale, and then other groups around the country, we're looking at individuals with epilepsy, people that were had intractable epilepsy, and they were being monitored for seizures as a way that they could potentially intervene. And while they were being monitored for these seizures, you could record directly from the surface of the brain. So what Greg and others were doing at the time is, actually Greg was interested at those times in language. And one of the things that they were looking at is letter string processing across the cortex and in the fusiform. And during that time, they had found so much responsiveness to faces when using other classes of data that Greg really started publishing a lot on that. I'm bringing this up because you're going to see some things that lead to a little bit of conflicting results when considering fMRI versus considering EEG. These are data I actually collected in the 1 year that I was doing intracranial recordings, which was now 14 years ago. So these are quite old, but the data, they're nice. They make an interesting point. One is that you can find various levels of specificity across the cortex. Here you can see this electrodes down here showing these face-specific responses. One here is the C5 electrode, which is right here. A1 electrode, wherever I pop that sucker right here, is actually showing a letter-specific response there. And you can see this in this early 200 microvolt potential along the cortex of the brain. And again here, this is from a different patient now. You can take a different sort of screening task, and here you have animals. You have Fourier transform, just scrambled images to see if there's any kind of visual consistency in there. You have faces, fruits, tools, and again, letter strings. This would be what we would consider a face-specific response. This N5 electrode here, you can see a big response here, and this J2, big response here to faces, but not the other stimulus types here. And again, it's not exactly one spot. There are sort of patchy organization where there seems to be some specialization for this closer into the occipital area, and again, up here in the medial temporal area as well. Functional localizers in fMRI work are mostly concerned with identifying this type of activity in the same way we might identify it with it in this intracranial EEG, and then applying sort of a convenient heuristic that allows for comparison. So by calling something the fusiform face area, I can then go forward and say, okay, now I have a map, and then I can map it on each individual subject, and then I can test the specific properties of it. I can test what the impact of attention is on it. I can test the impact of affect, maybe facial expression, and things like that. So it's useful in that way. One of the negatives or drawbacks of it is that sometimes people think that it would reify function, oversimplify sometimes complex brain functions to say something like processing of a face is all carried out in one area, which is not the intention. And just a note there at the end is just because you find activation for a particular class of a visual stimulus doesn't mean you've found the center of a complex function. And this is something that causes great argument for people that were both in field that looked at neuroimaging as being sort of too reductionistic, and people outside of fMRI as well. So you have this sort of back and forth between this really strong desire to be able to map what is a system of functional specialization without trying to boil it down to an oversimplification. So there's always been a little bit of a tension between the two. Now, what I want to show you from data that I had collected, what you would typically see in an fMRI study, which I said gets to be a little paradoxical or maybe a little conflicting at times. So here you have results of a general localizer analysis. In this case, I was showing faces, flowers, and headless bodies. Now I want to show you first this results when you look at the group level of faces versus flowers, which here you can see in the triangle. You get this strong activation, faces greater than flowers. It's stronger on the right side than the left side, which is something that we've seen with face perception over time. And then medial to that, you'll see flowers being active stronger than faces. This is a classic result that I could probably get in any number of subjects very simply just by putting them in the magnet, and we can demonstrate this. If we were doing a simple experiment for students in a class, I'd probably do something like this. Now, when you actually look though, and you use these headless bodies, and then you use those as a stimulus class, you find, again, this sort of lateral body is greater than flower. Medial, you don't get as much difference between the flower and the body as you did before with the face. And so it sort of leads you to wondering what's the difference, the processing demand differences for this unique stimulus class. And then the final part of that I want to make this point of is here, when you compare faces versus bodies, now you no longer have this specialization. So this area that you might call the fusiform face area, when using this second class of stimulus, a body without a face, no longer comes out in this group-based analysis. And then medial to that, you actually do have activation. Again, it looks like the body is maybe being treated as both object and face. There's different ideas as to what that meant. So some people speculated that maybe there was a contextual filling in of information. And others said, well, maybe there's also body-specific responses and that fMRI isn't resolving it in a spatial way right next to there. Go back to the intracranial data now. And again, as I showed you before, you have this faces versus and flowers. So the same thing, same basic experiment, but recording straight from the source of the fusiform in this case. Here you have this face response, and then you have this flower response, and you get almost nothing to the flower. You get this really nice N200 to faces. Now you throw in this body, this headless body, and again, this is flat. So the same thing that I showed you in fMRI, where these two stimuli seem to be equivalent in the bold response, is clearly showing a distinguishable difference on this area of the fusiform at 200 milliseconds. And again, here you have faces versus flowers in this occipital type of electrode. You throw in a body, and again, the body stimulus looks very much like the flower stimulus here. Again, great functional specialization for a face response at 200 milliseconds. Again, here's the, I think I showed you that before. Okay, yeah, those are just repeating, sorry. And here's another electrode here, just showing the same exact thing. So again, I can show you a couple of these, but these are all coming from different patients, but the pattern remains the same. We can find functional specialization for faces in the fusiform at 200 milliseconds, and we can resolve this in ways that we're not easily resolving with the fMRI signal. Now, sometimes when you study the brain, you hear about complementary sort of techniques. One being high in spatial resolution, meaning fMRI, the other being high in temporal resolution, ERPs coming from the scalp. EEG and ERPs coming from the surface of the brain are high in both, obviously, but it's very rare that we can do it because we're looking at people that are in for neurosurgery, so it's only a very tiny, small subset of the population. But to sort of sum up what I'm getting at here, you have this N200 component, which is showing little or no habituation to repeated faces. So what we've done over time is we've tested properties of it, and you can show that this early N200 that is doing a nice job distinguishing is pretty much insensitive to almost every psychological manipulation you'll throw at it, whereas these fusiform activations are pretty much influenced by every psychological manipulation you throw at it. So you can manipulate the attentive qualities, you can change the affect, you can change the emotional context, you can do all kinds of tricks with things that are task relevant or task irrelevant, priming, learning. Lots of things seem to change this response here. Very little changes the response here. So in general, the nature of the measurement is different, and the inferences you could really be making about this need to be limited by this. And it speaks to the idea that this again is not necessarily a center of face processing that is capturing all the various domains of what we do with faces. Okay, I already said that. All right, so face perceptions is something that I have sort of started to look at way back when, and I've been thinking about it a lot again lately. I'm bringing this up, and I'm kind of going through this long introduction to get back to a point that I'd like to make, which is that face perception and face memory itself is a really old problem in cognitive psychology and cognitive neuroscience. We have a lot of work outside of the domain of neuroscience where we have looked at how people remember faces, people's perception of faces. We can interrupt people's perception of faces by inverting them. People study things such as the other race or the other age effect, the tendency to remember people's faces that are more like those that are in the environment in which we grew up or developed in. And so there's just a lot of work in this domain. And we also understand from the context of neuropsychological work that there's disorders where people have trouble with faces. Prosopagnosics don't recognize familiar faces. People with autism have some interesting, sometimes deficits in both face detection and face recognition. And it's become a big work in cognitive neuroscience because of this specialization. It's been one of the most concentrated on in terms of functional localizers and category specificity in the ways that I described before. Okay, so, but here's the problem. Even if we can reasonably quantify a neural signature of face perception, does that tell us anything about how well one remembers faces, right? So even if we could find something that we would agree upon, a biomarker, this is the activity that we're going to look at for when someone is viewing a face. And even if we can agree upon what the principles of that are, which I said would be hard anyway, because it depends on how you're measuring it and with what tool you're measuring with it. Even if we can do that, does that tell us anything about how well one remembers?
Isaac's chair while we're eating lunch.
Sorry, I'm trying to mute people. All right. Even if we can do that, so the interesting thing about face recognition is that when they first started describing deficits in it, people thought, well, you either recognize faces or you don't. We do have documented cases of damage where there are individuals that have clear deficit that's associated with damage. And then when they started looking even further, there's also people that have clear deficits with no damage or no known damage. And we call these people developmental prosopagnostics. They struggle with faces. Really a lot with the advent of the internet, researchers like Brad Duchesne, who's at Harvard or at Dartmouth now, a lot of people have looked at face recognition across the population, and it varies. Most people, expert, you can definitely say, because faces are something that we do great recognition with, but there are some people that are better than others. And there are some people that are really bad at it. There are people that have deficits in it, prosopagnostics. And then on the other side of that, there are people that are excellent at it. They're exceptional. They're called super recognizers. They're rare. Again, they represent a tiny little portion of the tail of the population, but they recognize all faces they've encountered. It's kind of, it's intriguing how well they are, how good they are at it. So it is a spectrum of a capability. When they've taken this apart further, looking at this in twin studies are a good way of looking at the genetic component of a skill. So here in monozygotic twins, you can see twin 1, twin 2 performance on face recognition, highly correlated, 0.7. So clearly a argument for that face recognition, biological and genetic, heritable in this case. Dizygotic twins, on the other hand, they do show a significant association. 0.9 is not trivial in this case, but they don't share the same exact genes as their twins do, and it's less. So you can make this really strong argument that face recognition itself is heritable. Okay, so, sorry, let me move on here. So given that, let me go back to that in a second and just get my point across. So given this, we have a behavior that we know is variable in the population. We have strong evidence for biological genetic input that should explain some of these differences. And the differences themselves are likely stable, and they show a pretty good degree of stability to my knowledge when tested across time. So all that said, we still can't do a very good job of collecting neuroimaging data and explaining this variability. If I just collected people in the magnet, took all their activity in their fusiform face area, predicted how well they would do on this task, the results would not be terrific. They might be significant, but the effect size would not be great. So there's a couple of problems there. And these problems have been highlighted in recent criticisms of neuroimaging work in general. This summer, there was a lot of action on Twitter that I saw, which keeps the same flavor of every couple of years people start in on this, which is that fMRI, for all of its terrific utility in terms of mapping the brain at a group level, has not led to reliable biomarkers for disease or disorder to the extent to which we can count on them for telling us much about health and ways of designing interventions, ways of using diagnostics. So we still have a long way to go. In fact, a lot of the argument about it is pretty grave in terms of the way they talk about it as it's just a failure. But our argument is a little different. It's that there's a couple problems here. One is the way in which we encountered these types of problems to begin with. When fMRI first began, we were interested in inference at group levels. We were interested in brain maps on average. The way that we would use the things like the general linear model was meant to reduce variance and find congruence across participants, not to highlight where the differences are and then look at those differences themselves. At the same time, we also had big, super loud machines that are uncomfortable to sit in, and sometimes crappy stimulus presentation devices and things like that. And our tasks weren't great. So one thing that drives psychologists nuts is when you look at tasks that have no reliability, and then you're trying to use these tasks as stable markers of difference. So a lot of the tasks that we've grown accustomed to in MRI research are not psychometrically solid in this case. So in a study that we're doing in my lab right now is aimed at a potential solution to this problem on two sides. One is improved behavioral tasks. So this is my student Andrew Graves up in the corner there. He is working with another student in the cognitive area, Jesse Grabman, as well as working with some of our quantitative faculty on a technique here. This is exploratory graph analysis, which has been developed by Hudson Galino, which helps to essentially create tasks. They're working to create a task that is matched in structure and performance. So a face recognition task that's short enough to be in the magnet. It can be matched in structure and performance, both with the non-face stimulus and the face stimulus. The faces and control stimuli have to have a high similar split half reliability, so their performance across the task need to be reliable. They need to be short, and they need to be able to have a face and a non-face feature that have different abilities, so that you have ability to engage the different systems, so that you can have a dissociable memory task for the faces versus the non-face stimuli. And this is something that you can get with things like exploratory graph analysis in this case. This is something that we're doing on that end. On the other end, we're getting away from, as I said was a problem, this idea of trying to quantify single regions and then assuming that those regions carry the variance that can explain the individual differences. As we know, this isn't a great way to explain individual differences, because the ways that these types of areas were defined to begin with were often based off of areas where we find differences at a group level. So what we've kind of moved to now as a field, and I'll have more on this in a second, is looking at the connections across the brain. So we're trying to find measures that will be sensitive to individual differences. And a lot of the promise has come from measures of the connectome itself. So here, this is just showing you quickly that Andrew has built a model that I'll tell you a little bit more about how you go about doing this. But essentially, you can model the connections across the connectome. You can distinguish individual subjects from one another. And then you can look at variability within these connections and how well that variability maps onto a cognitive or a perceptual domain of interest. So a lot of this and a lot of our inspiration from this comes from some of the reports like this one here. I think this is a particularly seminal one by Finn and colleagues called functional connectome fingerprinting. In this case, what they've done is they've taken data from the Human Connectome Project. And in this data set, you have participants that have participated in resting state scans. You have participants that have taken part in attention, tasks of attention, tasks of social perception, tasks of emotion, tasks of self-control, generally an entire battery of things that cognitive neuroscientists might be interested in. And what they did is they had these 6 imaging conditions over 2 days, and then there's 2 resting conditions. They took 126 subjects. They took the brain and they break it out into 268 nodes. So you create 268 by 268 connectivity matrix. So what you could do then is you could take every one of those nodes connected with every other node. So you just take a correlation of them if you'd like to. Each element of the matrix represents an edge. So it's the connection strength between the two nodes. So in graph theory, of course, we're talking about edges in this case and the strength of those connections. And then what you can do is you take a database matrix with the highest correlation of edges with the target selected. So what happens is, for instance, in a REST1, REST2, you take all the REST1 scans and you train a classifier. And then this gets used to picking out where all the variability is, and it has something unique about each one of those subjects. And then after doing that, you can use that to predict who's who in REST2. So if you have some degree of a fingerprint, meaning there's some uniqueness about an individual's connectome, then machine learning in this case should be able to pick up on it. And indeed it does. So REST1, REST2 success rate was at 92.9%. So 93% accurate this model was at predicting identity of the participants. REST2 to REST1, so you just do this opposite, train on one and then look at the other set, was 94.4. We've done this with our own work. So this is from the Human Connectome Project, the Fin work that I represent here. Our own work, we looked at this in the VCAP participants. The VCAP participants are a unique group. This is from the Virginia Cognitive Aging Project. These are individuals that are 60 to 80 years old. We scan them as well. They have a little bit different scan procedures. They were scanned 9 minutes apart. They did do a demanding attention task in between the two rest phases though. So there probably should be some difference between them if we're going to pick up on it. But again, we see similar sort of results in this case. When we take REST1 to REST2, we had 94.5% accuracy, and REST2 to REST1, we had 96.74% accuracy. And again, this was with 92 subjects. If you break this down further, here's what it looks like when you look at a connectome. They look messy-ish on screen. They don't look like the pretty blobs of cognitive neuroscience work or for functional category specificity. What this shows you in this case, this is a tool that Finn and colleagues used as well in their original paper, which we found helpful for our paper, or for our project here. We have this differential power measure. This differential power measure indicates which one of these edges are being used the most to show unique variance. So which one of these edges are most informative when picking out which person is which, when helping identify the ID of each subject. All right, so these are, you can think of these as being the most sort of variable. And indeed, what they really found is the strongest sort of connections that show the most variability that can be used in the model are coming from prefrontal areas. This is frontal parietal here based off of the atlas we used. But these prefrontal connections and the prefrontal and parietal connections in general, both were very indicative of, or they both had a lot of the variability in it. And they also showed in a secondary analysis that they were also the best at explaining a cognitive variable like intelligence. And in the second measure, phi here, these are connections that are very ubiquitous across the sample. So they're in everybody, but they're not terribly diagnostic in terms of separating ID from one another. So they're connections that you would find, they're stable connections within subjects, but they're not highly variable across participants. And here you can see this is a lot of within network stuff, within visual cortex and such, connectivity within the networks themselves. And again, here, default mode, very strong across left to right, et cetera. Okay, so that's one potential solution, is better stimulus, better analysis tools. Another potential solution that we've been using is to think about ways that we can maximize variability. So not only do we have the problem in general category specific functional neuroimaging that we're optimized for looking at areas of congruence, we're also oftentimes looking at healthy subjects, in my own work, healthy adults 18 to 30, and processes of interest aren't terribly variable in that case. Yes, people are different socially, but they also have all kinds of different compensatory mechanisms and other things that are at play. One thing that we can do is try to capture the system during known periods of time where these functions change. And if we can do that, that might give us some insight into the various brain regions that are changing over time and the various brain regions that are showing the most variability. This is just to show you a figure that we've used this is actually from a grant itself. Basically, we've started from a Brain Institute grant run by my student, Amalia McDonald, to assess a pediatric population. And what we're really interested in this population is the emergence of prosocial behaviors at an interesting time as these kids enter school, as they go from young children into the early years of school. And we're interested in looking at various measures that we can capture from them, including fMRI and including a lot of behavioral testing. Some of this stuff we do not have data on. They're things that are more long-term ambitious goals. Some of them we do have some data on, some early data on that I'll show you in a little bit. So we have a long-standing sort of project there. And one of our overarching hypotheses in this project is to look at the role of a candidate biological system. In this case, it's the oxytocin system. And the reason why we've done this is really for a couple of reasons. One is that we know the oxytocin system is very important for the emergence of social behaviors. We know from work of my collaborator, Jessica Connolly, her students and postdocs and scientists, that the developing human, the way that they interact with the environment, and the way that they develop in their environment has a lot of interactions with genetic and other situational factors to influence the development of the brain and support this social perception. And we think that this is being carried out with at least some role from the oxytocin system. Some of our work over the prior decade in adults, most notably a lot of work from Megan Puglia here, who's in the Department of Neurology at UVA now, and she worked in our labs during the time we had run most of these projects, looked at the role of the epigenetic control of the oxytocin system. So the actions of oxytocin are mediated by one receptor type, the oxytocin receptor gene. I'm not going to talk a lot about that today. I can give a whole talk on that. But essentially, what you could do is you can capture epigenetic variability in this system, which governs the control of the endogenous oxytocin system, and we can relate it to a lot of neural endophenotypes. This was a figure from one of Megan's papers. Allison Jack, another postdoc who's now, or another graduate student who's now faculty at George Mason, she did some of the early work as well, looking at some of these measures too of social perception. And we have a series of studies that have essentially looked at this system and the way that we can represent epigenetic variability and how it plays out at the level of the brain. This is Jessica Connolly. She's my primary collaborator in this. She's the one that basically discovered the sites along the oxytocin receptor that show the most variability. They're the most diagnostic for classifying various differences, social differences. And also she has a complementary program in the Prairie Bowl where they could actually manipulate early social experience and show the impact on social behaviors themselves. And there's a lot of results that come out of that work too. So again, in this case, we have a nice candidate region supported by one receptor type coded on one gene. We have some inference that it does impact variability in key brain areas for social perception. And now we're trying to track it through development. I won't talk about this either, but I'll put a key plug in there. Again, work that Megan had done and continues to do now is to try to track this very early in life in infants. So we had a grant with the NSF to follow infants from 4 months of age and then follow them at 3 different time points to understand how their social environments, how their interaction with caregivers impacts the neural signatures that Megan has developed, such as neural entropy measures, and how this is related to differences in oxytocin receptor methylation and how oxytocin receptor methylation is changing over time, and whether or not we can develop this neuroepigenetic signature that predicts behavior, and whether this signature can be used as an early marker of dysfunction, which is ongoing work that Megan is working on with collaborators in the medical school now.
Are you guys ready?
Oh, sorry, let me go on from there. Okay, so a couple of issues that come up here and how we're solving them currently. And this is more related to going back to as the kids get a little bit older with developmental pediatric populations, but it does impact infants as well. One of the things that we have some issue with is that we need reliable biological markers and measurements. A lot of the early epigenetic work was done with blood. Children don't like getting their blood drawn. We can't ask parents to do it. We won't get any volunteers. We've been able to develop saliva assays as well. This is quite an old slide now. I just pulled it earlier and I didn't have the updated one. There's hundreds and hundreds of people. Megan's tested a lot of this. Amalia has tested a lot of this. Katie Kroll, postdoc in Jess's lab, has done a lot of work on this. In general, our saliva assays work, which helps for this type of these types of procedures. Another problem is that children have trouble staying still. This is an issue with functional neuroimaging, and this is a really big issue with connectome studies. One of the things you find in connectome studies is that one of the biggest predictors of connectome effects is head movement. So there's a lot of spurious results that come out of head movement, and particularly if you start getting into developmental disordered populations, this could really become a confound because degree of impact in terms of the degree of deficit might also really positively correlate with someone's ability to even keep their head still, which can create some issues. This is an image of a brain from a kid moving their head. This was actually my son moving his head. It looks like a picture where someone doesn't stay still because that's exactly what it is. And it causes big time problems with connectivity studies. What we've done to remedy this is we've developed a mock scanning protocol. This is from our old mock scanning environment, but we do have a newer one over at Fontaine now. Same system, just a different room there. You make it a nice, warm, welcoming environment, as Molly has done here. And you get the kids to learn to stay still in the fMRI, MRI environment. You play them movies, you teach them to play games where they keep their head still and they can keep their head within a little small box. It just teaches them to be able to stay still while you're playing magnet sounds and you're basically simulating the environment as best as possible. This head coil is the same. It's not the same because there's nothing in it that is technological. It's just plastic, but it has the same specifications in terms of size and fit as the head coil that we use when we actually do imaging itself. This is a figure. This is an older figure as well. Amalia has done this on close to 80 kids now. And this is just to show you that having an effective mock scanning procedure can really lead to you having really great results with head motion. Kids that are taking part in various tasks, this is a functional task. This is a task where they're actually asked to watch various video types that we've made of animate displays. This is from Inscapes, which is a technique that's been developed by another group, which is basically just it looks like 90s screensavers to me, but it's just to put something on the screen when you're collecting resting data so that you theoretically stay more calm and you have something to do, something to watch in that case. But there's not any kind of real psychological content to it that would be that engaging. Another thing that we do that I would talk about briefly in a minute is we run free viewing paradigms where kids are watching movies. Kids aren't great at doing psychological tasks all the time because they find them boring and they don't care that we need good results. So one of the things they are good at is watching movies though. And so they're entertained by watching something like Finding Nemo. In this case, we can have them watch these videos and then we can use some analytical techniques that can go about looking at various brain networks while they're watching these videos and that differentiate the content types. One of these techniques that we've been using is something called intersubject synchronization, intersubject correlation. It has a couple different names. It's really a technique that goes back to 2004, 2005. Yuri Hassan, who's a professor at Princeton, Dr. Malik there, also was involved in this type of work. And what they had done is they took participants and they put them in the scanner and they had them watch the movie The Good, the Bad, the Ugly. All right, and what that did for them is that they had this movie with really rich content. That's an old Clint Eastwood Western movie for people that haven't seen it. It has a lot of scenes, has a lot of faces, has a lot of changing dynamics to it. And at this time, no one had done anything like it. But what they were able to do is they just took every subject's voxels across their brain and did a bunch of pairwise correlations. And they showed something that probably shouldn't be all that hard to believe, which is that people's brains look remarkably similar when they're viewing the same content. So there's this synchronization. So two individuals' brains will look very much alike while they're watching the same content. Well, over time, researchers have adopted this to identify how are different content types treated. So I did some work back in 2012 with John Haidt, trying to understand positive emotions, which are hard to elicit in the brain, using basically Oprah videos to show how people represent sort of these positive regarded feelings for others. And now it's becoming an interesting tool for researchers looking at kids. Essentially what you get is you get a voxel time course from each voxel. So you get a time course of activity from each voxel as someone's watching a video. I'm not going to show you this video right now. This is from Finding Nemo. You have consistent across viewers, you have idiosyncratic, or you have spontaneous. So you have three different types of signal, really, when it comes down to it. You have those that are very similar across viewers. You have some that are just sort of spontaneous noise fluctuations. And you have some that are idiosyncratic, meaning that they're kind of reliable, but they're just their own sort of subjects viewing of that video, just their own sort of response from that area. What Cantillon and others did is they did this in children, and they did this both in kids and adults. And what they were interested in is the maturity of the cortex itself over time to these video types. And what they had is kids watching Sesame Street, and they created these maps that they called neural maturity maps. And what they could do is they could go children to adults. They could say, here's where adults are congruent to one another. Here's where children are congruent to other children. And here's where children and adults are the same. And here's where children and adults are different. So you could have this and you just simply line up subject from one to subject all the way down, subject one to all the way down. And then you can compare sort of where the spaces are that children and adults are maximally different. And then you could use this as a map of neural maturity. And they did, and they were able to come up with this metric, this neural maturity correlation. And then they were actually able to then look within a single area, this is Broca's area, and determine whether or not this neural maturity index maps onto a behavior. So in this case, a language type of behavior in children, which is changing during this time. And they had great success doing this. So we've been doing a similar technique with our studies, with our kids. We have them in the magnet. We have them at rest. This is from an older slide here showing children watching inscapes, and you run the intersubject synchronization analysis, the intersubject correlation analysis, whatever you want to call it. In this case, you have 23% of the voxels are correlated, and it's all coming back from these occipital areas. Again, as I told you, there's these sort of moving things on the screen. You are activating the visual system in a synchronized way because they're looking at the same visual content. You have this Nemo control. 42% of the voxels are correlated, so a little bit more of the brain. Again, you're getting more content area, more cognitive type of area. They're processing what's going on in the scene. In this Nemo social condition, this is a condition where the fish are working together to escape Darla, the fish killer in the dentist's office, for those that have seen Nemo. And in this case, you're bringing in even more recruitment of the brain because it's more rich social content compared to this control video itself. So again, we did the same sort of procedure that Cantillon and others did. You find the intersubject synchronization, we do the correlation matrix. We've tested this with mentalizing capabilities. We've tested this within the mentalizing ROIs themselves. So just taking areas from the Neurosynth online database and just looking at certain voxels that the field have described as being involved in mentalizing processes. And then doing this analysis between children and adults to determine whether or not we can get a neural maturity index. In this case, we're just using older kids and younger kids. Just to show you really quick, one of the highlights of what I think we're getting out of this, which I think is interesting, and some of this has changed as we collected more kids, and we're kind of in the middle of doing a lot of this. Amalia has also given the parents this SSIS, which is a social skills improvement scale, which measures problem behaviors and social skills. And it's a parental report in that case. Results from this are really intriguing. What you get is when you look at social skills and you look at average ISC values to all other children, in females in particular, you see a strong correlation in such that those that score higher on the social skills look more adult-like. So they look like the average of the older kids than those that score lower. So instead of using age as a predictor of change over time, you're actually using skill as a predictor of age over time. On the other hand, this is the SSIS ASD score, so problems, behavior, so social skill in terms of being a detriment. Again, you see less problems overall with higher levels of, or I'm sorry, better synchronization with lower scores on this, which mean better social behaviors. And again, here with self-control, better self-control with more of this mature brain response to NEMO, and the better brain response to NEMO, you get less externalizing behaviors. So again, this is a nice negative correlation in that case. So again, what we think we found here is a way of, instead of just looking at differences that are a result of age, we've actually been able to find a unique individual subject measure that's pulling out differences in phenotype of the kids. So whether that's a social characteristic as reported by parents, or whether that's something we can measure in the lab. And to the degree to which we can optimize this and do this better, we can learn more about functional systems supporting these behaviors. We can leverage this variability to understand the development of those areas, to understand how those areas support various behaviors, and to understand how those areas are involved in deficit, and what other kinds of biological, genetic, et cetera, and other factors that might influence that as well. So that's what our ultimate goals are in that case. I'm going to go ahead and open up to questions because I'm weary of time. It's 9 minutes to the hour, so I'll let Jack go ahead and start in on some questions now.
Jamie, thank you so much for sharing your work with us on individual differences. This is fascinating stuff. It's such a rich area. I can see why you're so interested in it. A couple of questions from me. The fusiform face area has been very famous or infamous, depending on who you talk to. And there's been a lot of work in looking at, obviously, faces versus other stuff, because faces are sort of special, that we believe. But there's also been studies which have shown that there's almost like expertise elements to the fusiform face area. So for example, people who are really interested in birds or antique collectible items or flowers or cars or something can often activate the fusiform face area. And it's given rise to the notion of sort of an object-topy of that area of the inferior temporal lobe. I'm curious about your thoughts on individual differences there, as in contrast to faces with, say, other objects, in particular, like the work of Isabelle Gauthier on some of her work on training people and stuff. You're very familiar with that, I'm sure.
Yeah, I mean, so one thing that's very characteristic of humans is that we are, first of all, we are very plastic neurobiologically, which allows us to learn new skills, develop new hobbies. Something like, let's take like a bird watcher, for example. You can go from knowing nothing about birds, and if you're passionate about becoming a bird watcher, you can, in a number of years, distinguish between 600 bird types probably. You have these big, brilliant brains that can develop hobbies and expertise for various areas. Now, to the degree to which visual specialization takes part and how that plays a role in these plastic capabilities and skills is a question that has come up in the face perception world, such that, are we just good at faces because we're expert at faces? Are we active in the fusiform? Do we show this response because faces are just a common area of expertise amongst the whole population? As I said before, faces are important to us. There's something that we develop over time. There's something that we get really good at. There are arguments all the way into the womb about the degree to which babies that are in the womb, shining lights into there can depict and differentiate between face-like stimuli, how early this develops, critical periods that it won't develop, and expertise for objects that does not follow that. And there are arguments back and forth. I'm not going to take a strong stance on that today, except for to say that it's possible, because neither one of those activation types can necessarily explain how good the behavior is in either way, then maybe this is, again, a case where the connectome can help us solve that. Can we find differences for the degree to which face recognition itself can be explained by functional connectivity differences? And how does this map on to expertise? And one natural way we could do this is we could test the connectome of people that aren't experts in something. We can make them go become experts in something, and then we can see if we can spot any of the connectome changes and whether or not those share some of those positive edges with face recognition. That would be a super cool experiment. It's an experiment that was done with GLM and blobs and on the fusiform, but maybe there's time for a modern update of that, and that might actually settle some of the critique. Other than that, it's an interesting debate. I'm not going to go too much further down that road of that, except for to say, I think that's another thing that data science could lend some answer to in an empirical way.
How might one quantify the degree of memorability of faces or other objects in a way, that one might try to do this kind of experiment, in the sense that, the person who last served you Starbucks has a face, but you probably don't remember what it was. But on the other hand, Madison Avenue seems to have figured out that face memorability can be linked to, products we might like to buy. And so there must be something about that. Is that something we can quantify? Something that how does that done that might help us with some of that uniformity that would allow us to make people experts in things? Do you know what I mean?
Yeah, certainly there's some argument of that. I mean, so everything about face recognition is interesting, and because you mentioned that in that way, is that outside of neuroscience, it's a super interesting question in terms of AI and technology. I mean,
yeah,
obviously at some point in time, Apple must have invested, some very, very large sum of money so that we could all look at our eyes and they can unlock themselves, right?
Yeah,
you know what I mean?
sure.
So face recognition is a problem that goes across many disciplines. And yes, as you said, I'm sure Madison Avenue has been interested in this for years. What's a memorable face? In terms of ways that people have done it in psychology, you could do it in a single session. How good are people that look at faces that are computer generated, so there's no chance they've ever saw them before, and then recognize them 20 minutes later? That's one way. That's a short sort of recognition timeline. So is there differences there, and are those meaningful for something? And then you have famous face tests, so people born in a certain year should have some X number of faces they would recognize, because you can have culturally relevant faces in there, and those will differ based off of how old you are. Is there a metric there? And there is. And and then there's arguments about, okay, is that longer term famous face different than the shorter term face recognition task I did? And the ultimate solution to that is that it really looks like there's some stable differences in both of those types of experiments. But they're dependent upon people's experience with faces. They're likely dependent upon the person's genetics and capabilities that they're coming along with, as I mentioned from some of those data from the twin studies. But it's a consideration, and it's a consideration when you test these things of what is it that you're interested in, things that people have encountered in the past, or people encounter in the present, or people making these sort of quick judgments. And like, I saw this person yesterday, and it seems like it's tapping into a common framework, but there are subtle differences likely.
First, this leads me to that in some of your NEMO task and some of the other tasks you described for us today, you discuss things like the exploratory graph analysis, the use of support vector machines, and some other types of machine learning type things that are now becoming just so popular. What other kind of data science related computational methods are you interested in seeing for the work that you're doing?
Well, yeah, I'm really interested in two things, and it's sort of colored a lot of what we've been doing lately. One, I am interested in things like Hudson's exploratory graph analysis and other researchers that have done things like this, because this is, I think, is a huge role of data science in diagnostics. So we do things like, for instance, Tim Salthouse, who was an authority on aging research at the University of Virginia, has published some of the most relevant work in that over time. What he would do is for years, he would take these people from the community and he would give them hours and hours of cognitive tests, bring them in 3 times in the summer, do a total of like 9 hours of tests, just an awful lot of testing, so that he could create very stable latent variables of processing speed, intelligence, verbal ability, cognitive capability, et cetera. Mm-hmm. There's a premium though, there's a problem with that. One is that for special populations, they might not come in for 9 hours, right? So, especially in aging populations, things like that. we need to get quicker. So we need to be able to have better factor analysis type of procedures, ways that we can reduce, where are we getting the maximal amount of variance that distinguishes people and tucks into the psychological process that we're interested in. And that's where something like EGA comes in, some of the other procedures that people do. Super interested in that for autism. It's one of these diagnostic tools where it's not like there's a yes or no, my child has a deficit here or doesn't, it's really tested amongst a lot of cascade of factors. Can we find the core factors and reduce them to better diagnostic procedures? So that's one, and that would help with individual subject differences at the brain level. The brain, the part that I'm most interested in, is this observation. As I mentioned, the Finn paper, that was the thing that got me most excited about this years ago, was that there is some stable variation in the connectome, which is really cool. Because if there's if you know nothing else about research methods going into anything, the one thing that always comes out is that, in order to pick up anything that's a meaningful individual difference, you need reliability. And you need to retest reliability. Bold GLM imaging isn't great for this. It's reasonable for some tasks, particularly easier tasks, but it's not terrific. But if the connectome has some level of stability that and is variable, and that variability can explain cognitive and social capabilities, then that gives us a real chance. Now, the problem there is that connectome models of attention, connectome models of face recognition, generally, if we do this 268 by 268 matrix, which is just one that we use because that's one that they've used before, that's 17,000 comparisons we're making. And the model fit will often pull out 1,020 connections, edges that are needed to predict that model. What's that tell you about the brain? That's a whole lot of connections. So it tells you that the brain is a highly connected universe that is involved in this complex behavior. But we want to get further down. We want to get more granular. So you need more techniques in that case. As you know, Jack, one of the professors starting in our department and in data science in the fall is Teague Henry. And he's involved in a lot of trying to find local differences within network science that can help us reduce this problem.
Well, with that, Jamie Morris, thank you so much for sharing your work on individual differences with us today. Fascinating work, such a rich area, so many different things to try across, not only in developing children, but across the lifespan. Just fascinating stuff. Thank you again. Thank you, everybody who joined us today. Have a great weekend. We will not be having a data science seminar next Friday. We're taking a little bit of a spring break, so giving everybody a week off, but we will see you the week after. Thank you so much, everybody. Enjoy your time. See you later.